Now we're just going to go on a little more to talk about the hormonal and neural regulation of digestive functions. So we're going to start with swallowing, because obviously food has to get into the mouth, so this is the initial place where everything's happening. And so when you take food, how do we produce saliva? Well, chemoreceptors within the mouth, the mechanoreceptors within the mouth, they respond to the presence of food. And those afferent impulses go to the salivary center in the brainstem, and then the parasympathetic nervous system stimulates a watery saliva secretion. Remembering, of course, that we have the sympathetic nervous system. If that's activated because you're very, very nervous, for example, you don't produce watery saliva, you produce a thick, viscous saliva, which makes you feel quite dehydrated in the mouth. So if we start at the left-hand side of this diagram, we're thinking we've got pressure receptors and chemoreceptors in the mouth. These receptors are stimulated, and they go stimulate the salivary center in the medulla, so that's in the brainstem. You can also have the smell, taste, and thought, etc. food coming in through the cerebral cortex, also stimulating the salivary center in the medulla. But that's a conditioned reflex, okay? Because you might be getting that because of food that you like. So for me, for example, it would be Indian food. The smell of a beautiful curry just gets me producing lots of saliva because I'm just anticipating or wanting to have that food. Anyway, so you'll all have different things that really make you want to eat, and therefore the smell, the taste, or the thought of a particular food type will stimulate your salivary center in the medulla. And so in that simple reflex is what we're thinking about. So pressure receptors going to the salivary center in the medulla through the autonomic nervous system stimulates the salivary glands to increase salivary secretions. We're thinking about the parasympathetic nervous system, of course. And like I said, the smell, taste, and thought, that's a conditioned reflex, depending on what you actually like. So what is in the saliva itself? Well, the main things that we need to be concerned about in saliva, it contains salivary amylase, and that begins the digestion of carbohydrate into disaccharides, for example. It contains bicarbonate, and that helps to neutralize the acids in food, because most people are actually very acidic food, so that bicarbonate plays a very important role in trying to neutralize some of that, the initial acids, and then the damage that can do to the teeth, etc. It helps to moisten and lubricate food. That makes it much easier to swallow, but also for dissolving, for taste. You can only taste stuff when the chemicals are fully dissolved, and they can therefore bind to your taste receptors, the different types of taste receptors that you have. It has antibacterial effects, for example. So it has things like lysozyme, which are antibacterial in their nature. It also helps to flush food residue away to help keep your teeth and gums clean and healthy. There's no nutrient absorption at this point, and that saliva also helps to form a food bolus, which obviously then gets passed to the back of the throat and starts the swallowing reflex. So, and that's what we're going to talk about now. So the first phase of the swallowing reflex is voluntary, but all the others are autonomic. In other words, you can't control them. So if you just think about sometimes when you might have had a boiled sweet and it suddenly hits the back of your throat, all of a sudden your autonomic nervous system is trying to swallow that boiled sweet. And that's because you stimulated the automatic part of the swallowing reflex. So the first phase, when you form a bolus, so you chew it, you masticate it, you form it into a bolus of food. But as soon as you push it to the back of the throat and it hits those mechanoreceptors on the back of the throat, all of a sudden the autonomic system kicks in and the swallowing reflex begins. So when you've done that, your bolus of food is voluntarily forced to the pharynx, where it hits those pressure receptors or the pharyngeal mechanoreceptors, and that stimulates the swallowing center, and that's in your brainstem. And that's where we hit the involuntary phases. And the first part of that is called the pharyngeal phase. And this is where your swallowing center inhibits your respiratory center in the brainstem. So you can't breathe at the same time as swallow. You elevate the soft palate. So here we go, the nice little diagrams. And that elevates up to prevent a food from entering the nasal passages. The position of the tongue prevents food from re-entering the mouth. And the epiglottis is pressed down over the glottis to prevent food from entering the airways. So that's the first phase that's all being controlled from the brainstem, the swallowing center in the brainstem. We then move on to the esophageal phase, and this is where the pharyngeal-esophageal sphincter opens up. Foods can now enter the esophagus, and then you have these peristaltic waves sweep along the esophagus via the vagus nerve. And so that's where you're getting smooth muscle contraction and relaxation, contraction and relaxation, contraction and relaxation, which propels the food down the esophagus until it hits the gastroesophageal sphincter, which relaxes, and then the food can go on to enter the stomach. So how does that peristalsis actually happen as it moves down the esophagus? Well, basically you're getting contraction of the muscle behind the food bolus, so it's smooth muscle, remember, and this is stimulated by mechanoreceptors, where the food actually is. So in other words, it stimulates relaxation ahead of the bolus due to stimulation of inhibitory neurons. Now, sometimes your food gets stuck in your esophagus. We've all been there. And this stimulates mechanoreceptors, which immediately activate the enteric plexus, and strong contractions are stimulated to force the food along. This doesn't actually involve the swallowing center, nor are we aware of their occurrence, though we can feel that the food is stuck. And of course, distension also stimulates more saliva production to help with that lubrication. So this is just another image just to show that swallowing reflex and what's going on as we pass food bolus down the esophagus. So where we have our bolus, this is stimulating the mechanoreceptors, and that then causes contraction behind the food bolus to help push it down, and relaxation ahead of the bolus to therefore allow the food bolus to pass down. Then, of course, the food bolus is passed down to the next part, so we get contraction behind, relaxation ahead. Gets pushed down, relaxation, contraction behind, and relaxation ahead. So remember, we have inhibitory neurons that are causing the area of receptive relaxation ahead of the bolus. The food then makes it down to the lower esophagus sphincter, which opens because it relaxes, and the food and that secondary wave of peristalsis forces the food into the stomach. So here's a nice little simple review question for you. So have a little read, press pause, and then we'll go through the answers. So salivary secretion can be influenced by the parasympathetic nervous system. That is correct, of course, because saliva becomes nice, thin and watery as we're starting to help to digest food and form that bolus to have it nice and moist. The hunger and fullness centers are the hypothalamus. Yes, that is also correct. The sympathetic nervous system. That is also correct. Remember, your saliva becomes more viscous and thick. And the cerebral cortex. That is also correct. The thought of food can stimulate saliva secretion to a small degree. So now we're going to move on to the roles of the stomach. So the stomach has multiple roles. It helps to store your food so that it can therefore be emptied at an appropriate rate for digestion and absorption to occur. So in other words, we get full digestion and full absorption of nutrients. The stomach doesn't absorb food, but it can absorb things like aspirin and alcohol, for example. The stomach also secretes things like intrinsic factor. So intrinsic factor is required for vitamin B12 absorption in the terminal ileum. It secretes mucus, which helps to protect the stomach lining from the acid that it also secretes. And it also secretes pepsinogen, which is activated into pepsin, which is a protease. So it starts to break down your proteins. We also have peristaltic contractions, of course, in the stomach. And they begin in the pacemaker region up in the top area, and they sweep towards the antrum. So if we're following this diagram down, those peristaltic contractions move down towards the antrum and towards the pyloric sphincter, which is the entrance point from the stomach into the duodenum, the small intestine. And each time we're getting that contraction, so we get contraction, forces a small amount of food into the duodenum, and then we get contraction of the pyloric of the pyloric sphincter, which closes it off. But because it's also closed off, it causes gastric mixing. So we get a nice regulated amount of food coming into the duodenum, and we get more mixing through these contractions and relaxation. So one of the main hormones released by the stomach is gastrin. And as you might guess from its name, gastrin is going to stimulate gastric acid, for example, cause the release of gastric acid. But when is gastrin actually released? Well, it can be released because of three different phases, if you like. So one is called the cephalic or the head phase. And this is when food is in the mouth. You might be thinking about or smelling or tasting or chewing or seeing food. This causes the release of a small amount of gastrin. You have the gastric phase. This is when the food is in the stomach. So the stretch within the stomach and the presence of proteins within the stomach will increase those gastric secretions. And finally, the intestinal food. So the food has moved from the stomach and it's moved into the small intestine. The initial phase is a very small amount of gastrin secretion, but after that, it's basically the inhibitory phase. And as food moves into the small intestine, overall, it helps to shut off the gastric juices as the chyme enters the small intestine. Because it's basically going, well, food's left the stomach now, it's come into the small intestine. We don't need as much gastrin and gastric acid and pepsin release, etc. Because gastrin, the hormone, causes the release of the gastric secretions, and the gastric secretions are your gastric acid, your intrinsic factor, and your pepsinogen, which goes on to cause pepsin release. Now, just so that you know, alcohol and caffeine, they can both stimulate acid secretion in the absence of food. Therefore, they should be avoided in people with gastric ulcers or gastric hyperacidity, for example. And protein is the most powerful stimulator of gastric secretions. And how is it doing that? Well, of course, your peptides and your amino acids, they bind to chemoreceptors, which then go on to stimulate gastrin release, which enters your bloodstream, which then goes on to stimulate the gastric acid, etc., release from the GI tract or from the stomach, more accurately. So what else does gastrin actually do? So it releases all these things, gastric acid and pepsinogen and hydrochloric acid, but what's it actually doing as well? Well, it enhances gastric motility, so smooth muscle contraction and relaxation. Stimulates ileal motility, same thing, contraction and relaxation. And it induces your colon mass movements. And all of those things together keep the contents moving through the GI tract when the new meal arrives. So it makes sense. New food arrives, we want to get the other stuff moving to get out of the way. It also stimulates mucus production in the stomach and the small intestine to help maintain the lining. And here's just a little diagram. Here's your capillary, here are your cells, and here is a nice mucus layer. And that mucus layer contains bicarbonate, and it's basically there to help to buffer against the very acidic gastric juices that are present in the stomach lumen itself. Whereas you can see pH is down at around about the 2 to 3 level. So if all that gastric juices could get to these cells, then of course they are going to get damaged. So we've hit the point where the stomach, we know what's going on in the stomach. We're starting to break down proteins because of the pepsin that's there, but we're not breaking anything else down at this point. So what is the state of the food and the fluid mixture, or the chyme as it's otherwise known, that's entering into the duodenum? Well, the carbohydrates may be partially digested into disaccharides by salivary amylase, because remember that started in the mouth. Proteins, these may have been partially broken down into small peptides because of the action of pepsin and hydrochloric acid. Hydrochloric acid doesn't do any digestion, it just denatures the proteins so that the pepsin has access to them. Fats are still mainly triglycerides. We haven't really done very much there. There's a little bit of lipase in the saliva, but not very much. And the acidic environment. So remember, it's very acidic in the stomach. So what's coming into the duodenum is also acidic. Now, this will inactivate the bile salts, which are required for fat absorption. And it'll also inactivate the enzymes that are going to come in to help digest our food products. So we need to neutralize that acidic environment in the duodenum. So that's the first thing we really, really must do. And so what happens is the acid in the duodenum binds to receptors because we've got a pH change, and that stimulates the release of a hormone called secretin. And so here, if we're looking on the right-hand side, acid in the duodenum stimulates receptors, which causes the release of secretin from the duodenal mucosa, enters the blood, goes to the pancreatic, so the pancreas, and in the pancreas, remember, in this case, it's going to be an exocrine secretion, and it causes the release of bicarbonate into the duodenal lumen, which neutralizes the acid in the duodenum, which is what we want. So therefore, the secretin stimulates bicarbonate secretion from the pancreas. That buffers the acid, bringing the pH to a more neutral level, and it also inhibits gastric secretion. So that's the other role of secretin, and we know that that's what's happening in the duodenum. It also stimulates the liver to produce bicarbonate, stimulates the liver to produce bicarbonate-rich bile, which, remember, bile is stored in the gallbladder, and it also stimulates the gallbladder to contract. So therefore, you're getting more bicarbonate entering the duodenum to help to buffer against that pH. But you're also getting the products of bile being released into the duodenum at this point. So waste products. So, for example, from waste products of red blood cell degradation, so that's your bilirubin, for example. You have cholesterol that's passed through there, and other waste products that are stored from the liver within the gallbladder. So what is the gallbladder? Well, it's a 9 centimeter long pear-shaped organ, has a capacity of about 50 mils. But actually the liver produces about 1 litre of bile per day, but that's concentrated by the gallbladder. Looking at where it is, it just passes, so it's very close to the liver, and your bile drains down the bile duct and into the gallbladder where it's stored. And of course, you can get gallstones in there, so you'll have heard of gallstones. So what's present? Well, you have bile salts, which help with fat absorption. You've got bilirubin, a breakdown product of red blood cells. You've got excess cholesterol. You've got both phospholipids, proteins, electrolytes. These are all secreted by hepatocytes and transported down the bile duct to the gallbladder where it's concentrated. And it's concentrated into a greenish yellow coloured liquid before being emptied into the duodenum. Remember, being secreted or gallbladder contraction occurring because of secretin release. So you can see that the gallbladder products, your bile passes down the common bile duct and then into the duodenum, the small intestine. Like I said, secretin stimulates bile generation, gallbladder contraction, and therefore bile secretion into the duodenum. So what else has come into the duodenum? We know it's got lots of acid in there, but also at the same time, we've got fats that are coming in there, and we've got proteins in there, and we've also got carbohydrates in there. But the fats and the proteins in the duodenum simulate CCK release. So that's another hormone, of course. And so now what we're thinking is, right, it's fat and protein that are stimulating CCK. So what is CCK going to actually do? Well, it's going to stimulate the release of proteolytic enzymes. So lipases, amylase, disaccharidases, and nucleases, and to help with the digestion of the food products that it's detecting. So again, you've got fat and protein products in the duodenum. CCK is released from the duodenal mucosa, enters the blood, goes to the pancreas, and causes the secretion of your pancreatic enzymes into the duodenum, so you can then digest the fat and the protein. At the same time, it decreases gastric motility and secretion. So again, it's another thing that's stopping those gastric secretions. But it does increase motility of the small intestine and the colon, therefore we're getting contraction and relaxation. So as you can see from this, it's fats and proteins that are causing the release of CCK, which is causing the release of the pancreatic enzymes. And it's interesting that carbohydrates present don't have any real effect on pancreatic enzyme secretion. And the proportion of the enzymes don't change with release. So we don't get an increase in peptide or proteases released just because we're having a high protein diet. But the amount of them does change. So the more protein you have, or the more fat you have, the more pancreatic enzymes are released. So in other words, it's dependent on the amount of the stimulus. Having said that, there is some limited evidence to suggest that long-term changes in diet may alter the proportions of enzymes made and therefore released. For example, you may get slight increased proteolytic enzyme release if a high protein diet is regularly consumed. But the evidence is fairly wishy-washy on that, so I wouldn't read too much into it. Just presume that a standard amount of H enzyme is released when you get that fat and protein stimulus. So really, what are the overall functions there? The overall functions of CCK are to allow sufficient time for fat to be digested and fat to be absorbed, but also carbohydrates and proteins, because we're releasing those enzymes as well, leading to carbohydrate and protein digestion. It also causes gallbladder contraction, so releasing bile salts and bicarbonate and bilirubin and other wastes, and it induces fullness through the hypothalamus. The last little enzyme we're going to talk about, hormone rather, is GIP, or glucose-dependent insulinotropic peptide. And this is, it has other names as well, so the names have changed over the years, just so that you're aware of that. And this is released because of glucose hyperosmolarity in the duodenum. So in other words, it's being released when it detects a change in osmolarity. So it's binding to osmoreceptors. And so it stimulates insulin release from the pancreas in anticipation of absorption of the meal. In other words, it's a feed-forward mechanism. So what's happening? Your nutrients are coming into the GI tract. You have an increase in the amount of glucose that's in there, which increases the osmolarity. That osmolarity stimulates the release of GIP into the blood, and GIP binds to a GIP receptor on the pancreatic beta cells to trigger the release of insulin, which is being released in anticipation of glucose entering the blood. Okay, so it's quite a cool little mechanism. So like I said, GIP is known as other things. The other common name used, just in case you read it, is gastric inhibitory peptide. But really what I want you to know is, or think about, do you think insulin secretion is greater when a glucose load is given orally or intravenously? So press pause, have a little bit of a think about whether you think insulin secretion is greater in each circumstance. Okay, so hopefully you worked this one out. It shouldn't be too hard, but insulin secretion is greater overall when given orally due to the effect of GIP on its secretion. Because, of course, you don't get that GIP secretion when you give IV glucose, because you're not passing anything into the stomach and the GI tract. So the last thing I want you to do is just to see what you can remember on those different hormones. So gastrin, secretin, CCK and GIP, what's the stimuli for their release? Where are they released from, and what are their key functions? So I'd like you to press pause and just work this out for yourself before we go through it. Okay, gastrin. That's released because through those three phases, remember, so when the food is in the mouth or you're thinking about it, when it's in the stomach, and when it's in the small intestine. So really we're thinking about the smooth smell, taste, thought of food, etc. Peptides and amino acids and distension in the stomach, and then to a lesser extent in the small intestine. Where's it released from? It's released from the G cells in the stomach, and it stimulates the release of gastric acid, pepsinogen, mucus, and intrinsic factor. Secretin, acid, is causing that release. It's released from the duodenum, and of course it functions to secrete bicarbonate, which helps to neutralize the acid as the food gets into the duodenum, bile production, and gallbladder contraction, because remember, bicarbonate's also in the gallbladder. So basically it's functioning in a large way to neutralize the acid that comes into the stomach, into the small intestine rather. CCK, it's peptides, amino acids, and fatty acids that stimulate its release. It's also released from the duodenum, and it causes pancreatic juice release, so all those enzymes that are going to digest your food, induces fullness, and causes gallbladder contraction. And then we have GIP. So what stimulates that release? Well, it's an increase in osmolarity, so glucose. It's released from the duodenum and causes insulin secretion. So those are the main summaries. Do remember that those hormones do also influence contraction and relaxation in the GI tract. So CCK causes motility in the small intestine, for example, and decreases gastric motility and secretions. So don't forget that we have those other roles as well.